Good morning, teachers. I am Anjali Yadav, pursuing BSc Zoology Honours from Maitri College. My roll number is 0140. Today I am going to explain a topic, that is Fasciola Hepatica. So let's move on and study about Fasciola Hepatica in brief. Fasciola Hepatica belongs to kingdom Animalia, phylum Platyhelminthes, class Trematoda, family Fasciolidae, genus Fasciola, and today we are going to deal with species Fasciola Hepatica. Fasciola Hepatica is commonly known as liver fluke or sheep liver fluke. Its body is leaf-like and dorso-ventrally flattened. Fasciola hepatica has suckers, the oral one which is smaller and the ventral one which is larger, and the larger ventral sucker is also known as acetabulum. Suckers are used effectively to anchor the parasite as they reside inside the bile duct. It is a hermaphrodite. Life cycle of Fasciola hepatica is around 9 to 11 years in bile duct. Respiratory and sensory organs are absent. It is cosmopolitan in distribution throughout sheep raising areas. It is found as an endoparasite in the bile ducts of liver of sheep and other mammals. The disease caused by the fluke is called fascioliasis. Liver fluke causes a disease known as liver rot, and it causes great economic losses in sheep and cattle as infected animals become anemic, which means the RBC's count is decreased and significant weight loss is also seen. Let's talk about hosts of Fasciola hepatica. There are two types of host, definitive host and intermediate host. Definitive hosts include sheep, cattle, buffaloes, human and other mammals. Humans are accidental hosts, that means they are not very common hosts. And intermediate hosts include freshwater snails from the genus Limnia. Fasciola hepatica has an indirect life cycle, that is, it has a numerous larval stages. So Fasciola hepatica has 7 life stages: egg, miracidium, sporocyst, radiae, cercaria, metacercaria, and finally the adult fluke. Here we can see the images of all the life stages. Let's study about life cycle of Fasciola hepatica. Imagine an adult Fasciola hepatica is present in sheep's body and produces eggs. These eggs are passed out via feces of the sheep. So now the eggs are out of animal's body. Once the egg reaches fresh water, the eggs start to develop and become embryonated in around 10 to 15 days at an optimum temperature of 23 to 25 degrees Celsius. Egg is oval, yellow-brown in color and operculated, which means it has a lid-like structure. So embryonated egg will hatch and produce miracidium. Miracidium has cilia all over its body. Miracidium is capable of living for only 24 hours. So the egg was in water and the miracidium produced was also in water. Miracidia has cilia which helps it to swim and find its intermediate host, which is snail. If miracidia finds snail within 24 hours, it gets inside the snail body, and if it is unable to find snail's body, it dies. When miracidia contacts the snail, there is no use of cilia further, so it loses its ciliated coat. Now, inside the snail, the miracidium develops into sporocyst. Till now, we saw that one egg produces one miracidium and one miracidium produces one sporocyst. Asexual reproduction takes place in snail's body and number increases tremendously. So one sporocyst will produce numerous mother radii and it will further produce numerous daughter radii, and daughter radii will give rise to various cercariae. So after 5 to 7 weeks of infection in snail's body, one miracidium gives rise to approximately 649 to 650 cercaria. Cercaria has a tail, so it helps in locomotion. Cercaria will now exit the snail's body and swim to settle down on some aquatic vegetation. Then it will lose its tail and form cyst around it. So it forms a new stage known as metacercaria. Now sheep and cattle roaming around by chance eats aquatic vegetation. So metacercaria enters their body. So metacercaria is ingested by the definitive host. In case humans ingest uncooked aquatic vegetation, miracidium enters their body and humans become accidental host. So sporocyst, radia and cercaria were developmental stages of Fasciola hepatica in snail's body in water, and metacercaria was present on aquatic vegetation. And finally, metacercaria enters into host body. Now, metacercaria will come out from the cyst in duodenum of host's body. Metacercaria will travel around in the body to reach their final destination, which is bile duct. In bile duct, metacercaria will develop into adult fluke. In humans, the time taken for Fasciola hepatica to mature from metacercaria into an adult fluke is roughly 3 to 4 months, which is 13 to 15 weeks. The adult flukes can produce up to 25,000 eggs per fluke per day. Eggs will start to appear in feces of host in around 13 to 15 weeks after infection. And then eggs are passed out via feces of the host and this cycle continues. Here is a complete diagram of life cycle of Fasciola Hepatica, starting from unembryonated eggs produced via feces of the definitive host, which develops in around 10 to 15 days to become embryonated egg in water. Then it hatches to produce miracidium, and miracidium swims and find out intermediate host, that is snail, and penetrate inside it. It will further develop to form sporocyst, then radii, then cercariae. Cercaria will swim, and after it settles down on aquatic vegetation, it loses its tail and form a cyst around it. Then metacercaria on vegetation ingested by definitive host get infected. Then metacercaria exist in duodenum and penetrate intestinal wall and migrate through liver parenchyma to biliary duct. There it fully gets mature and start producing eggs, which are passed out via feces, and the cycle continues. Here are references. Thank you.